Topic for presentation is DNA damage and repair. Many chemicals, ultraviolet radiation, they cause damage to the DNA in our body cells, and cells must continuously repair the damaged DNA. So one is excision repair. This occurs when any of over 50 repair enzymes remove damaged parts of DNA. And the enzymes DNA polymerase and DNA ligase, they replace and bond the new nucleotides together. See, if we see, the error is here, newly made strand and binding of mismatch proofreading proteins, and here one loop is made and strand is removed and repair is done and strand is corrected. Here, this is old template strand. It contains a methylated bases, so wrong base could be identified. In this, a portion around that area is cut and removed by an endonuclease, and the gap is filled with correct base sequences by DNA polymerase enzyme. Here the DNA ligase that is sealing the nicks. The cause is there may be the environmental factors, they damage the DNA, and chemical agents, they damage DNA. They are divided into 4 types. Abnormal regions of DNA, either from copying errors or DNA damage, are replaced by 4 mechanisms. One is mismatch repair, second one is base excision repair, third is nucleotide excision repair, and the fourth one is double strand break repair. Types of damage to the DNA are: one is single base alteration, that is by depurination and deamination of cytosine to uracil. Next is deamination of adenine to hypoxanthine and alkylation of bases, insertion or deletion of nucleotides, base analog incorporation. These are the single base alterations. Next is 2 base alteration. In this UV light that induces thymine dimer. Next is bifunctional alkylating agent cross-linking will occur. This is second one. Third is chain breaking. Ionizing radiation is one, cause radioactive disintegration of backbone element. Toxicity to free radical formation is another one. This is cross linkages, and between bases in same or opposite strands it may happen, and between DNA and protein molecules, that is example histones. Due to mismatch of bases, due to errors in replication, and chemicals like vinyl chloride, H2O2, aromatic hydrocarbons, they cause DNA adducts such as ethanobases, oxidized bases, alkylated phosphodiesters, and cross-linking of DNA. Then DNA repair mechanisms: nucleotide excision repair, mismatch repair, base excision repair, mutations, and double strand break repair. Regarding mismatch repair, here mismatch base pairs are corrected after replication. An extensive region from the mismatched area is a new strand. The new strand is removed and correct nucleotides are added. Here, single side strand cut by endonuclease. Here, defect removed by exonuclease. Next is defect repaired by polymerase in this step. And here ligation by ligase. Finally, the repair. Base excision repair, if you take, the depurination is spontaneous. Deamination of C and A are also spontaneous. And DNA glycosylases, the enzymes that recognize the base of alterations. The wrong base is removed by cleaving the N-glycosyl bond. Thus, purine and pyrimidine, the episites, are generated. The area around this site is then removed by epiendonucleases. These portions are resynthesized by DNA polymerase and then combined by ligases. See here, heat energy with this uracil DNA glycosylase enzyme, and nucleases are acting over here, DNA polymerase and ligase. Finally, repair. Nucleotide excision repair. This is used to replace regions of damaged DNA up to 30 bases in length. It involves more gene products than the two other types of repairs. This essentially involves the hydrolysis of two phosphodiester bonds in the strand containing the defect. A special excision nuclease accomplishes this task. Thus, a fragment of DNA from 27 to 29 nucleotides long is excised. See here the recognition and unwinding. Next, oligonucleotide excision by cutting it two sides, and resynthesis and religation. Double strand break. Two proteins are involved in the non-homologous rejoining of break, and they are KU and DNA dependent protein kinase. DNA PK allows for the approximation of the two separated ends, and the activation of this KU helicase results in unwinding of the two ends. The unwound approximated DNA forms base pairs. The extra nucleotide tails are removed by an exonuclease, and then the gaps are filled and closed by DNA ligase. See here, binding, approximation, then unwinding, approximation, unwinding, and then alignment and base pairing. Here alignment is taking place like this, and base pairing and ligation. And the mechanisms, the problems and solutions are a mismatch repair, copying errors, single base or two to 5 base unpaired loops, and here the solution is methyl directed strand cutting, exonuclease digestion and replacement. Next, base excision repair. Here this is spontaneous, chemical or radiation damage to a single base. And the base removed by N-glycosylase, A basic sugar removal and replacement is the solution. And nucleotide excision repair. This is spontaneous, chemical or radiation damage. Here the solution is the removal of an approximately 30 nucleotide oligomer and replacement. Double strand break repair. Here this is ionizing radiation is the problem, chemotherapy, oxidative free radicals, and solution is synapses, unwinding, alignment and ligation. And coming to mutations, also after proofreading, also some defects may retain in DNA. These are called mutations, though due to the change in the basic sequence of DNA, due to faulty replication or DNA repair. In a germ cell, line mutation rate is about 4 nucleotide changes per year. And this is associated with DNA repair mechanisms or xeroderma pigmentosa, defective mechanisms, sensitivity to UV light, skin cancers, and ataxia telangiectasia, defective gene, sensitivity to UV light, lymphoreticular neoplasms. And one more is Fanconi is anemia, that is the defect in DNA cross-link repair. Increased occurrence of cancers will be there in this. And another one is Bloom syndrome. Defect is in DNA ligase or helicase, lymphoreticular malignancies. Cocaine syndrome is another one. Defect in NER mechanism, transcription factor 11H is defective. This is nucleotide excision repair. That is defective, and stunted growth and the mental retardation will be there. Hereditary polyposis colon cancer. This is also known as Lynch syndrome. This is here the mismatch repair is defective. And the mutations or the changes in the nucleotide sequence of DNA may occur in somatic cells, not passed to offsprings. And the mutations happen regularly. Almost all mutations are neutral. And chemicals, UV radiation cause mutations, and many mutations are repeated by enzymes. Some type of skin cancers and leukemias, they result from somatic mutations. Some mutations, the types, chromosome mutations may involve changing the structure, loss or gain, and chromosome mutations, deletions, inversions, translocations, non-disjunction, and duplication. 5 types they exist. Due to breakage, the deletions, the piece of a chromosome is lost. See here, deleted genetic material is seen. Breaks in chromosomes are also seen. And inversions, chromosome segments break off, segment flips around the backwards, and segment reattaches. Here, reinsert piece of DNA in the inversion. See, breaks in chromosomes, inversions, reinserted piece of DNA. And duplication occurs when a gene sequence is repeated. See here, section is duplicated. Next is translation, involves two chromosomes that aren't homologous. And the part of one chromosome is transferred to another chromosome. And translocation is the thing here, the thing before translocation and after translocation, the chromosomes. Non-disjunction, failure of chromosomes to separate during meiosis, causes gamete to have too many or too few chromosomes, and disorders are Down syndrome, Turner syndrome, and Wolfert syndrome. See non-disjunction. Yeah, meiosis starts normally, and then one set of homologs does not, and meiosis second occurs normally, and all gametes have an abnormal number of chromosomes. This is original chromosome, and this is duplication, and this is deletion, and this is inversion, and this is also inversion. Gene mutations change in the nucleotide sequence of a gene may only involve a single nucleotide, maybe due to copying error, chemicals, viruses, or etc. Types of gene mutations include point mutations, substitutions, insertions, deletions, and frame shift mutations. Point mutation change in a single nucleotide includes the deletion, insertion, or substitution of one nucleotide in a gene. See point mutation. Sickle cell disease is an example, is the result of one nucleotide substitution. Occurs, see sickle in the hemoglobin gene. Frame shift mutation, insertion or deletion, one or more nucleotides. Changes the reading frame, like changing a sentence. Proteins built incorrect. Frame shift mutation, original one, the fat, cat, egg, and fat, like frame shift here. This is the example, like amino acid sequence changed here. Frame shift mutation. These are all the recording mutations, and what gene repair mechanism and damage. The damages, mainly due to free radical damages, UV radiation, and heat, such things. And we'll continue in the next class regarding the remaining topic of molecular biology. Thank you.